Malaria remains a significant global health problem with an excess of 1 million deaths per year, primarily among children under 5 years of age in sub-Saharan Africa. The Plasmodium parasite, the causative agent of malaria, lives a complex life cycle in two evolutionary divergent hosts, the human and the mosquito. Although Plasmodium transmission is clearly recognized as an important step in the parasite life cycle, very little is known about the sexual or gametocyte stages, which are the only parasite forms that can be transmitted from a human to the mosquito. Human infection is initiated by the bite of an infected Anopheles mosquito, wherein sporozoite stages are injected into the skin and then traffic in the bloodstream to the liver. Following liver stage development, thousands of merozoites are released into the blood to infect red blood cells, RBCs. A subset of these asexual parasites receives a commitment signal that initiates sexual differentiation, or gametocytogenesis. Merozoites are then released from sexually committed schizont-infected RBCs, invade a new RBC, and form the gametoring stage. Maturation, the final phase of gametocytogenesis, is initiated, and the gametoring enters a cellular differentiation process, divided into 5 developmental and morphologically discrete stages. The inner membrane complex, IMC, which is supported by the microtubule network, is involved in this process. During maturation, the IMC and the microtubule network expand around the periphery, thereby influencing cell shape. During stage 4, the IMC has been stretched and completely surrounds the parasite. When the microtubule network is disassembled, it results in the relaxation of the ends of the gametocyte, producing the characteristic stage 5 crescent. The Plasmodium parasite has completed gametocytogenesis. Terminally differentiated stage 5 male and female parasites circulate in the peripheral bloodstream and capillary venules and are easily distinguishable by light microscopy in Guim-sustained thin blood smears. Interventions to block gametocyte persistence in the bloodstream and transmission to mosquitoes are urgently needed, especially in the context of malaria elimination and eradication. Transmission-blocking vaccines, TBV. Following immunization with a TBV, antibodies elicited against parasite surface antigens can either clear gametocytes in the bloodstream or, upon uptake with gametocytes into the mosquito midgut during blood feeding, prevent downstream developmental stages, thereby blocking malaria transmission. Classical TBVs can target gametocyte, gamete, or ookinete proteins. However, of these 3 stages, only gametocytes are present in the human host, and targeting gametocytes may allow for natural boosting of the vaccine. To date, the majority of candidate gametocyte surface antigens remain untested as TBV targets. Vaccine efficacy depends on the intrinsic ability of a vaccinated individual to mount an immune response, and this is negatively impacted by the poor health and nutrition status of vaccinees in malaria-endemic countries. Therefore, transmission-blocking drugs offer an alternative approach that can circumvent this hurdle. Transmission-blocking drugs. Primaquine, PQ, is the only drug currently available that is effective against stage 5 gametocytes. The exact mechanism is not well understood, but is reported to affect parasite mitochondria. However, clinical use is limited by PQ's toxicity, particularly in individuals with glucose-6-phosphate dehydrogenase G6PD deficiency, which is common in malaria-endemic regions. Artemisinin is an effective antimalarial, but activity is limited to asexuals and stage 1 through 3 gametocytes. Discovery of new drugs that are focused on preventing gametocyte development, killing mature stage 5s in humans, or preventing gametogenesis and fertilization in the mosquito could result in significant reductions in the number of infected mosquito vectors. However, the underlying genetic and biochemical pathways driving gametocytogenesis, and as a result the number of potential druggable targets, remains unknown. In the era of malaria elimination and eradication, research must be focused on breaking the cycle of malaria transmission. To do so, we must fill the gaps in our knowledge about gametocyte developmental biology so that we can develop a multifaceted approach to saving lives and eradicating this disease.